We have developed ShinyArchR.UiO, an R-based Shiny app that facilitates accessibility and visualization of single-cell assay for transposase-accessible chromatin sequencing data in a user-friendly, interactive, and open-source web interface. Here we present ShinyArchR.UiO with an example single-cell ATAC-seq dataset for hematopoietic cells. App initialization will take a few seconds, and this can be seen tracked on the right-hand top panel. After the initial setup of ShinyArchR.UiO from our environment or command line, Launch Browser will open the app on your default browser. In the Single Cell Cluster tab, users can select and explore 5 different multidimensional reduction U-maps, plots side by side. Here each dot represents individual cells grouped before and after integration of single-cell ATAC-seq data with single-cell RNA-seq data based on different analysis. All the individual U-maps can be downloaded as PDF, PNG, or TIFF. In the Single-Cell ATAC-seq Peak Browser tab, users can select genes of interest and press Plot Track button to visualize single-cell ATAC-seq peak across the locus. The browser shows chromatin accessibility tracks grouped by single-cell ATAC-seq clusters or by original samples, and up to 500 kb of a selected locus can be explored. Peaks and coaccessibility between original samples or clusters are shown below the browser window. In the Peak to Gene Links tab, users press the Plot Track button for a selected gene to visualize the peak to gene linkage genome accessibility for the gene of interest, which correlates single-cell ATAC-seq with single-cell RNA-seq data. In the bottom panel, arcs represent coaccessibility for the peaks. The Feature Comparison tab shows multi-dimensions reduction U-maps, allowing users to compare genes of interest with 3 different matrices. Gene score matrix U-maps show predicted gene expression for a gene based on weighing insertion counts in tiles nearby a gene promoter. Gene integration matrix U-maps shows linked gene expression with the inferred gene expression obtained through gene score matrix. A sequence logo is represented from U-map with motif matrix. Here the full list of genome positions of motif can be downloaded as a CSV file. The Pseudotime Trajectory tab allows users to visualize pseudotime trajectory heat maps for 4 different matrices in 2 panels: Gene Score Matrix, Gene Integration Matrix, Motif Matrix, and Peak Matrix. The 6th tab shows peak to gene links heat maps identified across the whole single-cell ATAC-seq and single-cell RNA-seq datasets. This heat map may take a few minutes to plot, depending on the size of the data and the computational resources available. In the About tab, you can find out more about us, the scope, and the approach of ShinyArchR.UiO. We have presented ShinyArchR.UiO, an app that adapts pre-processed Archer data of single-cell ATAC-seq for intuitive genome-wide exploration and sharing to make the data available for a collaboration group or larger audience upon publication.